Hello, biology students. Today we are going to chat about X-linked or sex-linked traits or diseases. It really just depends. We're going to call them traits because the examples we use, they're not life-threatening, and disease has a kind of negative implication. But these X-linked or sex-linked traits have to do with sex chromosomes. So when we're talking about chromosomes, we're talking about DNA, and specifically in humans. And male and females, humans have 46 chromosomes, 23 pairs, and the 23rd chromosome for females, genetically female, they will have two X chromosomes. And then we use this little symbol here to represent female. In terms of the 23rd chromosome for a genetic male, that individual is going to have one X and one Y chromosome, and we use this little symbol here to represent a male. Now, when we're talking about these X-linked or sex-linked traits, these are traits that are going to affect males more than females. And that should be like, jump out at you and say, well, why are males getting affected more than females? Well, that has to do with the fact that males only have one X chromosome. So if this chromosome happens to be a chromosome that carries a particular trait, in this case, maybe a trait for, say, color blindness. We're going to talk about that in just a minute. That male doesn't have any other X chromosomes. It only has that copy, that gene for color blindness. So that male is going to be colorblind. Now, in the case of females have a little bit of a unique circumstance where we're going to talk about that below. So like I mentioned, we're going to be looking at color blindness for our example of an X-linked trait. And my apologies here as I scroll down. As we're looking here, some of the highlights didn't come through when I scanned. So I'm just going to go in now and kind of make those highlights as we go along here, and hopefully this works out well. Yeah. So this is another highlighted one, and then these are both highlighted. Right, and that'll make a little bit more sense as I go through. So here, we're going to be talking about color blindness, like I said, and in this case, in this example, we have an individual who is XY, and that should jump out at you as an individual that is male. And in talking and labeling, talking about these X-linked traits and labeling these X-linked traits, we use a superscript. So that means above, superscript. And in this case, I use the letter B for color blindness. Those individuals that are colorblind, there are a couple different types of color blindness, but they don't see colors, quote unquote, normal. They might mix up colors. So in an individual who has a capital B copy of the X chromosome, this individual has the normal or unaffected X chromosome. So this individual has essentially the good copy of the gene for color blindness. However, in this example, I used a little lowercase b and I kind of shaded it in. You can kind of see the B, the lowercase b there, but the shading didn't come out very well. So just kind of be mindful of that as we're going through here. So this individual has an X chromosome, but it has a little copy of the gene for color blindness, which is unfortunately the bad copy. This individual inherited the gene for color blindness. So the capital letter represents like the good copy of the gene, and the lowercase letter represents the bad copy for the gene. So this individual is still male. However, they have this particular disease or trait. And again, this is kind of, it's not very serious in terms of life threatening. Color blindness is not that serious. However, this individual is what we would call affected. Okay. Moving on to the females. The females are a little bit different when it comes to X-linked traits or diseases. So an individual that is XX is female. This individual has two capital Bs. Those are the good copies of the X chromosome. So this individual is normal. This individual is unaffected. Now, in this next example, this individual has two X chromosomes, is female genetically, has one capital B, and then one of these lowercase b's. This individual is normal because that capital B, that good copy, covers up the bad copy. So this female is not colorblind. However, they are what we call a carrier for color blindness. So again, this female is normal, they are unaffected, but this individual is a carrier. And that comes into play when we do some Punnett squares. In this last example here, we have two copies of the quote-unquote bad gene for color blindness, which means this female is going to have the disease. So the only way a female can have a disease or trait in X linkage that is inherited according to X linkage is if they inherit two copies of the gene for a disease or trait. And in this case, it is color blindness, and this individual, this female, would indeed be colorblind. So as we move along, I'm going to scroll down. I'm just kind of erasing my little highlights here. And I'm going to scroll down, and I want to show you an example. Okay, and follow along with me as we go through. So this says at the top, it's an example problem. Complete a Punnett square. Complete a Punnett square for a male with color blindness and a normal female. See, my highlights came out a little bit better in this scanned copy, but here is my male, and this male has color blindness. I see that in the word problem. A male with color blindness, And if I'm not sure, I can always go back to this kind of chart. And this chart doesn't really change for any of the X linkage problems. And then the female is normal. That means they have two good copies of the gene, two capital B's. So I set up a Punnett square. Here's my square. And it looks kind of crazy, but let's try to keep it simple as much as we can here. We put our X and Y across the top. Okay, for our male. And see this copy of the X chromosome has that diseased X chromosome. And we want to make sure we recognize that, so we want to make sure we have that little b. This is our female. On the left, both copies of the female's X chromosome are good, so capital B and capital B as a superscript. We fill in a cross, and then do the same thing here, following along with my pointer, and down, and then we bring the Y down and the Y down. Now, when we analyze X linkage Punnett square problems, it's a little bit different because we have to look at females. These individual offspring would be female. And we have to look at males. These individual offspring would be males. And in this case, both females are X with a capital B, X with a lowercase b, which means they're carriers, but they're normal. They don't show color blindness at all. And we would say 100% of the females are normal, but they are carriers. In the males over here, we see that the X chromosomes are both X with a capital B, that's that normal X chromosome, and then the Y. So 100% of the males are unaffected or normal. So we can also look at this in terms of the total offspring. We could say that 50% of the total offspring are carrier females, 50% of the total offspring are unaffected males, because 50% are female, 50% are male. We add 50 plus 50, we get 100% offspring. This is really a cool problem just to look at. On a side note, this mother here is unaffected, but the father is colorblind, yet none of their kids could possibly be colorblind. Okay, any daughters could be, are going to be carriers, but will not be affected, which is kind of neat. I hope you found these problems interesting. The next video is going to be another example of an X-linked trait or sex-linked trait called hemophilia.